Welcome to this explainer. Today, we're diving right into the most sophisticated, incredibly complex manufacturing plant in the known universe. I'm talking, of course, about the human cell. We're going to frame the cell as this highly regulated microscopic factory, and we're going to track a single molecule of glucose, our primary raw material, from the second it hits the outer security gates all the way down into the fiery engines that literally power our existence. It's a wild, fascinating process, so let's just jump right in. Here's our blueprint for today. We'll start at the gates with membranes and cellular transport, then decode the electrical wiring of nerve impulses. After that, we break down glucose in glycolysis, fire up the mitochondria, and finally, check out our alternative backup fuels. All right, part one, membranes and cellular transport. These are the factory gates. Our factory is secured by an asymmetrical lipid wall, and it's constantly being maintained by these specialized worker proteins called flipases and flopases that literally just move lipids back and forth. Now, take a look at how this place regulates who comes in and who goes out. A uniporter is pretty straightforward. It allows a single species to cross via passive diffusion, just flowing with its natural concentration gradient. Easy, right? But when the factory needs to move things against that gradient, it has to use coupled transport. You've got your symporter, which moves two species in the exact same direction, basically using the energy of one to drag the other along for the ride. Then you have an antiporter. This couples the downhill flow of one species to push another in the completely opposite direction. This acts as either primary or secondary active transport, depending on the situation. Moving on to part two, nerve impulses and signals. Believe it or not, our factory relies on its own built-in electrical wiring to process messages from the outside world. Let's see how that electricity actually works. So if you remember one thing here, it's this number, -70. Specifically, -70 millivolts. This is the resting membrane potential of a typical nerve cell, meaning the inside is relatively negative compared to the outside. How on earth does the cell maintain this? Well, it's constantly running this active sodium-potassium pump. Think of this pump like a frantic, tireless night watchman constantly bailing water out of a sinking boat. It burns ATP energy to aggressively shove 3 sodium ions out of the cell, while only bringing 2 potassium ions back in. This uneven 3-for-2 exchange creates this incredible electrical tension that's absolutely necessary for a signal to fire. And when it does fire, here is the exact sequence of that action potential. We start at our resting state of -70 millivolts. A stimulus comes along and opens ligand-gated sodium channels. If we hit the threshold, bam, voltage-gated sodium channels burst open. Sodium absolutely rushes in, and the cell depolarizes, spiking all the way up to +40 millivolts. Almost immediately after, voltage-gated potassium channels swing open, allowing potassium to violently drop out of the cell. This drastically drops the charge, hyperpolarizing the cell all the way down to -100 millivolts, right before the pumps reset the whole shebang. That cascading wave of voltage changes, that's exactly how a signal races down the wire. Okay, section 3, glycolysis. This is our main assembly line, the place where we actually start extracting energy from our raw materials. To really get glycolysis, we first have to understand the type of work we're doing on this floor. Glycolysis is a textbook form of catabolism. This is the process of breaking down large, complex molecules into smaller, manageable subunits. It's an oxidative process, and the key takeaway here is that it releases energy. We are literally taking the chemical bonds of our food and shattering them to harvest power. Just as a quick counterweight, let's mention anabolism. This is the exact opposite. You take small molecules and build them up into larger structures. It's a reducing process, meaning it requires an input of energy. Now, while anabolism is how our factory builds its machines, for our glucose molecule today, our focus is strictly catabolic. We want to tear it apart. You're going to see 2 vital types of enzymes doing the heavy lifting on the factory floor, kind of caught in a constant tug of war. Kinases are the enzymes that take a phosphoryl group from an ATP molecule and aggressively slap it onto our substrate. This either energizes or destabilizes the molecule. On the flip side, phosphatases are the enzymes that come in and remove those phosphoryl groups. Throughout our catabolic journey, pay attention to the kinases. They are the main tools manipulating our glucose molecule. All right, welcome to the cytosol floor. This is the entire glycolysis assembly line. Notice how it's basically split into 2 distinct halves? The top half is the energy investment phase. We actually have to spend 2 ATP just to prime the glucose molecule and split it in half. You gotta spend money to make money, right? Well, it pays off. The bottom half is the energy generation phase. Those 2 halves are processed into 2 molecules of pyruvate, generating 4 ATP and 2 high-energy NADH molecules in the process. When you subtract our initial investment, we net 2 ATP. Now, glycolysis is a 10-step process, but the factory manages this entire line at just 3 irreversible checkpoints. These are all controlled by those kinase machines we just talked about. Step 1 uses hexokinase. Step 10 uses pyruvate kinase. But the absolute most critical checkpoint is step 3, which is controlled by phosphofructokinase. Let's just call it PFK. Think of PFK as the factory's incredibly strict shift manager. If the factory's overall ATP levels are running low, PFK screams at everyone to speed up the conveyor belt to meet demand. But if energy is high, it just takes a coffee break. It halts the assembly line to save on resources. Let's move deeper into the core. Section 4, the mitochondria. This is the main engine where we generate maximum power. So we netted a measly 2 ATP from glycolysis, but we still have those 2 molecules of pyruvate left over, and they are holding massive potential. To tap into that, we transport that pyruvate deep into the mitochondrial matrix. Once it's inside, an enzyme complex called PDHC converts our pyruvate into acetyl-CoA, releasing a little carbon dioxide along the way. That acetyl-CoA gets tossed into a spinning wheel of chemical reactions you probably know as the citric acid cycle. The rate-limiting bottleneck here is the conversion of isocitrate to alpha-ketoglutarate. But here's the real secret of this cycle. Its primary goal isn't actually to make ATP directly. Sure, it spits out some GDP, but its main job is harvesting these extremely high-energy electron carriers, NADH and FADH2. You really need to treat these things like highly volatile batteries. And, just like the assembly line before it, high levels of ATP and NADH act as a feedback loop to just slow the spinning wheel down when the factory's fully charged. Next up, we take those highly volatile battery carriers over to the inner mitochondrial membrane, right into the electron transport chain. You can picture this as a highly coordinated super-fast bucket brigade. Complex I and II rip the electrons away from NADH and FADH2, handing them off to a mobile carrier called coenzyme Q. Complex 3 then grabs them and passes them to cytochrome C. Finally, Complex 4 takes these now exhausted electrons and hands them safely over to oxygen, reducing it to water. As these electrons are passed down the line, protons are violently pumped out, creating a massive pressure gradient. But listen, this incredible system has a massive fatal vulnerability. Saboteurs like carbon monoxide and cyanide are lethal because they specifically block Complex 4. They prevent the electrons from ever reaching oxygen, which causes a devastating factory-wide backup that halts all power generation literally in its tracks. But, assuming no cyanide is present and the system is running smoothly with plenty of oxygen, we arrive at the ultimate payoff of aerobic oxidation: 30. That is roughly how many molecules of ATP we get per single glucose molecule. That massive proton gradient we just built up rushes back into the matrix through a magnificent molecular turbine called ATP synthase. As the protons flow through the F0 transmembrane channel, they physically, mechanically, spin the F1 head inside the matrix. This forcefully binds ADP and phosphate together to just churn out massive amounts of ATP. It's an incredible piece of microscopic machinery. Which brings us to Section 5, alternative fuels and recycling. Because things don't always go according to plan, right? Let's see how the factory handles emergencies. What happens when the glucose actually runs out? Well, the factory turns to proteins. It takes their carbon skeletons and breaks them down into amino acids for fuel. These amino acids basically fall into 2 camps. You have glucogenic amino acids, which can be repurposed into precursors to synthesize brand new glucose. And then you have ketogenic amino acids, which are broken down to produce ketones, like our old friend acetyl-CoA. Actually, a fun fact here, only leucine and lysine are exclusively ketogenic. Everything else has some glucogenic properties. But there's a catch. Burning protein has a cost. It produces these toxic amine groups that turn into ammonia. The cell's solution? It just ships the ammonia out of the factory entirely, sending it over to the liver where the urea cycle safely packages it up so you can dispose of it in your urine. Okay, here is the master blueprint that ties this entire tour together. You can see glucose entering at the top there, flowing through glycolysis to become pyruvate. If oxygen is present, everything runs perfectly. It flows straight down into the citric acid cycle and oxidative phosphorylation. Those are our massive aerobic engines. But what happens if the oxygen supply is suddenly cut off? What if our factory is suffocating? The system adapts. Pyruvate gets shunted to the left, straight into anaerobic glycolysis. An enzyme called lactate dehydrogenase swoops in and converts it into lactate. What this does is clear the backup and regenerate NAD+, which allows that basic glycolysis assembly line at the top to just keep churning out a tiny but absolutely life-saving trickle of ATP. That lactate is then shipped out via the Cori cycle back to the liver to be recycled.
As we wrap up our tour of the cellular factory today, I really want to leave you with a provocative thought based on what we've just learned. We know now that the electron transport chain is brilliant, but it is terribly fragile. If a toxin like cyanide can shut down our entire cellular power grid just by blocking oxygen from receiving electrons at complex 4, how might future medical science engineer alternative final electron acceptors? Could we find a way to keep the factory running during toxic exposure, or even during a stroke when oxygen is cut off? The answers to massive questions like this literally lie in the blueprints we've explored today. Thank you so much for joining me on this deep dive into the microscopic factory. Keep wondering, keep exploring, and I'll see you in the next explainer.